Our next speaker is going to be Dr. Adam Foster. He's a serial oilseed pathologist at the AAFC Charlottetown Research Development Center in PEI. Dr. Foster and his team have worked on numerous industry-related research projects over the last 6 years with collaborators across the nation, both regionally and national, for different research goals. His research includes cereal breeding line screening for FHB and powdery mildew residue resistance, surveillance of FHB causing species in the Maritime provinces, and the study of agronomy practices for disease control in cereals and soybeans. In the lab, his team is studying the effects of residue management on pathogen load for Fusarium, FHB forecast model development, the effects of crops, the effects on crops of the abundance of plant pathogens in the soil microbe, and is developing techniques for editing genes of fungal pathogens. Thanks, Adam.
Thanks for the introduction. Taking masks off with glasses is always fun when you loop them around the handles. I am Dr. Adam Foster, the serial and oilseed pathologist from Charlottetown, PEI. And this presentation also contains data from some of our collaborators. A big person who did a lot of work on this is my graduate student, Emily Johnstone. And actually, as we speak right now, she's in the greenhouse spraying wheat and barley plants with Fusarium spores and characterizing the differences in the Fusarium isolates for the virulence. Dr. Justin Renaud has done mycotoxin analysis for us from AAFC in London. And I work on some national projects with Dr. Kelly Turkington, who's a really fantastic pathologist out of AAFC Lacombe. And I've included some of his data here too, because we're running some trials for him, and I'm skimming the data off to do more disease forecast model evaluation with it. And we've got support for a lot of our disease surveys through a project with Brent McCallum. So today I just want to talk about two major research objectives. I think my bio was like way too long. Like really I work on like 3 different things. And this is like one of the things that I work on, and these are 2 of the objectives. The first objective is that we're doing right now is to evaluate the pathogen populations that are causing Fusarium head blights in the Maritimes. If you look at the Canadian plant disease surveys going back across the country, we know a lot about the populations of Fusarium pretty much in every single part of the country, except the Maritimes has been kind of a bit of a black hole for about the last 20 years. Now, doing these surveys takes a lot of time and effort, and it takes a lot of samples, but it's really important because once we actually collect the isolates, we can start studying them, and we can start finding out which fungicides work best to control them, seeing if there's differences in virulence, and this is the base information we need in order to make new management suggestions. The second objective I want to talk about today is our efforts to find the best Fusarium head blight forecast model for the Maritimes. Fusarium head blight is a really important disease across the entire world, and really good scientists have developed all sorts of different forecasting tools in all sorts of different environments. But none of them have ever been tested and validated in the Maritime region, and this is what we're trying to do using real-world data from on the ground. This is kind of a little bit of a FHB that I found in the field this year on barley. I'll be honest, in our Harrington research trials, we didn't find a lot on barley, but we found a lot of infection on spring wheat. And this is one of our 5-year rotation trials that's led by Dr. Kelly Turkington. So before I get into the survey work, I just wanted to give a bit of a background of the life cycle of Fusarium. If any of you heard me 2 years ago, this is the same slide, and I'll try to be as quick as I was back then. Fusarium head blight actually starts with the resting structures of the Fusarium fungi that are called Parathecia. These structures actually live in the residues of previous crops, especially corn. Like, big explanation point. Pathogen absolutely loves corn. When the conditions are right, these Parathecia actually explode with literally the greatest measurable force in all of nature to knock these spores way up high into the air, where they can travel really long distances. But most of the spores actually don't make it really through the wheat, and then just kind of get stuck on the plants there. When they actually hit anthers. This is where they can cause problems because they actually infect the anthers, and from the anthers they move into the seeds, and that's where they'll start forming Fusarium head blight. And then after the infection starts to set, you'll start getting these orange spore masses called sporodokia. If you see these orange masses, that's just a mass of spores. And what ends up happening is when it rains, these spores actually just splash from one plant to the other. And this can actually cause a secondary spread of infection within the cycle, within the field over short ranges. Then at the end of the year, what ends up happening is bits of infected material go back into the ground, and these parathecia basically develop, and then they overwinter, and then they wait for the next crop to continue the cycle again. In pathology, we like to use the concept of a disease triangle. I think with COVID, we all kind of are starting to understand what it means when we say you need to have a host, and you need to have an environment, and you need to have a pathogen. So for COVID, you have the COVID virus, the host, that's us. The environment, well, that's what, any closed room where we're all in. For Fusarium head blight, it's the same kind of thing. We need to have a pathogen. Those are the Fusarium species. We need to have the environment conducive to disease. And this is basically having a high humidity, lots of rainfall, and moderately warm temperatures. Fusarium likes it pretty warm, but it actually tends to get suppressed by the really hot temperatures if you get into like India and Brazil. We normally don't get those temperatures here though. And you need to have a host, and in this case, this would be cereals, and cereals when they're at their susceptible heading stage. So now I want to move on and talk about our Fusarium head blight surveys, which is all about trying to uncover the mysteries of the top of that pyramid. So this past year in 2021, we did 2 surveys. The first survey we did with the Atlantic Grain Council's on-farm agronomy work. They submitted us samples from all 3 provinces in the Maritimes, and we plated them just like this, where we get the seeds, we put them on the media, we pluck out all the individual molds that are starting to grow, and we isolate them into pure species, and then we use some molecular techniques to be sure that we're identifying the correct ones. My team also did a local survey in PEI of wheat, where they just traveled to a few fields, took some pictures, got some visual assessments of leaf disease, or of leaf disease and FHB, and brought some samples back in the lab so we could confirm that what they were seeing was actually FHB. So what are the big species of concerns? What are we actually looking for? Well, there's a whole bunch of different Fusarium species that can cause Fusarium head blight, but in Canada, the one we're most worried about is Fusarium graminearum. And the reason is when it infects grain, it contaminates the grain with deoxin of alanol. And deoxin of alanol is a mycotoxin that can cause all sorts of problems for humans and animals. But there's about another 16 species that can potentially cause Fusarium head blight. And in Canada, we commonly see Fusarium comorum, cerealis, Sporotrichoides, Poe, Equiseti. Each one of these species also produces its own mycotoxins. But the reason these aren't considered as important is they don't normally do as much damage to grain. They don't drop as many of these mycotoxins in the grain, so they don't contaminate it as much as graminearum does. But that said, if you go to different parts of the world, some of these other pathogens are actually the worst ones. So the on-farm agronomy fields are approximately in these locations. I don't have the exact GPS site locations, but that's approximately where the field sites were. And for the survey results, well, disease levels were overall moderately high this year. I say moderately high because we didn't see large amounts of Fusarium in every sample, but we did see a large amounts of Fusarium in a bunch of samples from all across the region. In total, we plated about 2,600 seeds, and 10.6% of them we were able to culture a Fusarium culture out of. And out of that, 277 of them were Fusarium graminearum. So we cultured a lot of Fusarium graminearum. We also did mycotoxin analysis from all the provinces. And just a rough overview, we found that most of the DON we found was actually in the samples that were from New Brunswick and, Nova Scotia in wheat and barley, followed by PEI barley, then New Brunswick barley, and the lowest amount of DON was actually found in PEI winter wheat. We didn't have any PEI spring wheat samples in this survey. So what species did we find by crop? Well, the most abundant species in both of the crops, wheat and barley, was Fusarium graminearum. And the populations were actually relatively similar. Second most abundant was Sporotrichoides, and the third most abundant was Avanasium for both. And we also saw Fusarium Poe pop around a lot. Now, if we look by province, well, PEI had Fusarium graminearum as the most abundant pathogen, but so did New Brunswick and Nova Scotia. It was the most abundant in all 3 provinces, and actually this is the first time since I've been doing surveys, since 2017, where I've actually seen Fusarium graminearum as the most abundant pathogen in all areas, in all crops. So that really shows that at least since 2017, this was the year with the highest level of Fusarium head blight pressure. In the past 2 years, I think Aaron kind of pointed out really well that the 2020 year was a really dry year, and we got a lot less Fusarium head blight. That being said, though, we still managed to culture actually a lot of Fusarium. In fact, the same amount of seeds were contaminated in 2020 as they were in 2021, but they were contaminated primarily with the pathogens Fusarium spirotrichoides and Fusarium poe. And when we did mycotoxin testing on these samples, we actually did find T2 and HT2 toxin, and we even found a little bit of nivalenol. But it wasn't anywhere near a guideline limit, and it would just kind of confirm that, yes, these are present and they are contaminating grain with mycotoxins, but they're not causing the same amount of damage. But the other thing we found is that Fusarium graminearum was still popping up. Not in every sample, but in every province, in both the crops, we got a few Fusarium graminearum isolates. It means that even in the driest year we've ever had, really, the Fusarium graminearum will stick it out. It'll find a crop and it'll still cause infection. So we've got all these Fusarium isolate. What's our next step? In the last 5 years, I've picked up about 500 Fusarium graminearum isolates, and we've started actually testing them for fungicide sensitivity. Essentially what we do is we fill microtiter plates with media that contains different fungicides at different concentrations. Then we put a little plug of the fungi on those plates, and then we measure over time how fast it grows, and we compare that to where there's no control. We don't have any results to show for this yet because we just started doing that in the last month. But so far, it looks like we are starting to see some differences in between different isolates. And one of the differences we just see in this isolate here is you can see this one little isolate right here has absolutely no color in it. It's actually hypersensitive to this fungicide. We're also doing greenhouse trials on a selection of these isolates to see if there's differences in virulence, and that's what Emily's doing today. So I'd like to move on now to talk about Fusarium head blight forecast modeling. Fusarium head blight modeling is just one part of Fusarium disease management. Modeling helps make decisions on whether it's necessary to apply fungicides to control or suppress the disease. But it doesn't replace good agronomy, having resistant cultivars, and particularly having a good rotation and managing corn residues. Continuously planting any cereal crop can result in this pathogen kind of building up in the field, and then when you do get bad conditions, it just makes everything worse. But when we're looking at Fusarium head blight forecasting models, we're taking the data from the disease surveys, we're taking weather data from Environment Canada weather stations and skimming whatever we can off of other weather stations online. And we're also applying, running tests where we're applying fungicides based on the risk predictions of those models to see how accurate they are. The final goal, what we're trying to do, is produce something that looks like this. This here is the risk map, Fusarium head blight risk map that Manitoba growers have right now. This summer, they can log into a website, say which crop they want, and it'll give a risk for the entire province, color coding where it actually is. Even better than that, the Americans actually have the Fusarium head blight prediction center that's run through their scab awareness form. And this is so incredibly detailed that you can put all sorts of information on your crop in to even customize the risk forecast to you. This I like. So how are we doing this? Well, we decided to do basically take Environment Canada weather stations from across the Maritimes. For today, I'm going to make a few demo maps, and in the demo map, I only picked one weather station per agricultural region as defined by StatsCan. I don't know what that means actually, but it does nicely break out the map into a color pattern that I could use. It does mean, though, that some weather stations like PEI are certainly closer together, though, than the ones that are in New Brunswick. On a final model, we'd want to have as many weather stations as possible to actually have as much local detailed data for risk predictions. So Our testing of these models is still ongoing. We still have to basically crunch the numbers from our field samples, and we've just received our dawn data analysis that we're working on. But we did test a bunch of different models, and every single model we tested is measuring the fusarium risk at enthesis. Our early results found that pretty much the models that looked at relative humidity and temperature were the ones that were giving us the most accurate responses. Whereas the ones that were looking at rainfall were really highly variable. Unfortunately, rainfall isn't really a uniform measurement that you can get in one area. For instance, it can rain in Charlottetown and it doesn't necessarily mean it rained in Harrington. So if you're going to use rainfall models, having rainfall measurements as close to where you're taking the data as possible is important. We also found that any model that used weather forecasts was really highly dependent on the accuracy of the weather forecast, and weather forecasts in the Maritimes are not always the most accurate. So overall, we found that there was different models that were good for winter wheat, spring wheat, and barley, and that's not surprising. These are different crops, and the disease actually forms differently in these crops. These models were approximately 70% accurate at predicting the epidemics, with about a 16% false positive. So that would be saying there's risk where we didn't see it on the ground, or an 8% false negative. That's where disease levels were high, but the model didn't indicate risk. That's actually what I'm more worried about, and what we're trying to work on now is modifying the models to actually minimize these false negative suggestions. So how will this all work? Well, I'm working right now to develop a web tool that will essentially download weather data over the last 7 days from a bunch of different weather stations, use the weather station data to crunch the mathematics of the model, and then spit all that out onto a map that could be hosted onto a website that a grower could look at on their computer or on their phone. A simple tool that would just basically tell what the risk is, just like a weather forecast. So here's some examples that I produced from our 2021 data. Now, the examples that I'm just showing in this next series, these are just the wheat models. The barley models are a little different, but when I had 20 different maps all in a row, I had a hard time keeping track of which one I was actually looking at. So the first risk date we chose to look at here was June 18th, and this is because this is the first date that we saw our winter wheat going into enthesis at Harrington. And on this date, the model would have predicted that actually risk was either very low in Queens County or low in most of the Maritimes and high in southern Nova Scotia. One thing you'll notice on this map, it's always high in southern Nova Scotia. We picked the Yarmouth weather station, and it's like apparently always humid down there. Always, every day, no matter what, is humid. So we're going to work in the future to, pick a different weather station to represent the region down there. So if we move into about a week later, this is kind of getting into the end of when the winter wheat was starting to go into enthesis. Actually, the risk started to pick up. We had a couple of rainfall events. Humidity started to go up a bit. And then all of a sudden, PEI went from being a very low to low risk to a low to moderate risk. And if we move just a few days later, when barley started to go into heading, actually the risk started to drop down a little bit there as we ended up into just a little bit of a drier period, except for, of course, southern Nova Scotia that stayed incredibly humid. But 3 days later, we got major rainfall events over that period. And the risk for the entire Maritimes was detected as being either high or very high. It then stayed this way for about another week. And then it started to actually decrease a bit down to a more moderate risk. And then for our last day, this is our late planted wheat trial, we found the risk for the entire Maritimes was either high or very high. And the reason for this is we actually had warm temperatures at night with high humidity. Tropical nights, they call them, where the temperature doesn't drop below 20 degrees, but it stays incredibly humid, are one thing we should worry about when it comes to FHB, because fusarium really does most of its growth and attack at night, and it really likes warm temperatures and humidity. Yep. See, Yeah, that's not something I've actually looked at, but that's something we actually pay a lot of attention to in our FHB nursery, where we actually inoculate, and we're essentially inoculating to screen breeding lines for resistance. One thing we know is that there's a real sweet spot in when we get our harvest in. If we leave them in the field too long and we give Fusarium a chance to just keep growing, the dawn does increase to the point where all of a sudden moderately resistant cultivars won't look resistant anymore. It's definitely a factor too that's been studied in other regions, and I think it would hold true for here as well too. There is the adanthesis conditions though, or what is when you would apply fungicides. So this is why we're targeting guidance at this point. Because if you did wait until August conditions to apply fungicides, that's off-label, that's too late, and it might do more harm than good. To validate the trials, we actually conducted 7 research trials at the Harrington Research Farm last year. These were in fancy Latin square designs, so we can try to minimize the variability in the field plots. 1 trial was for winter wheat, and 3 trials each for spring wheat and 2-row barley. The spring wheat and 2-row barley trials, we staggered planted. The first planting we got in April 16th, which props to my team. That's the earliest we've ever got anything planted, so I was really proud of them. Our mid-planting was kind of more, I think, would be more of a typical planting period, the second week of May, May 5th, and our late planting was getting near the end of May. End of June would be kind of end of May and beginning of June would be kind of where this is at. And we tested 3 treatments versus an untreated control, Prosaro XTR, Miravis, ACE, and a biocontrol that my grad student pulled out of the ground in, actually, Aaron Mills cover crop project a couple of years ago. And all the sprays were timed for wheat at around 50% anthesis when we could, and barley at full head emergence, and they were all applied at the maximum label rate. So first for results for winter wheat, I'll start with yield. There was no treatment effect on yield, but unfortunately our winter wheat had a bit of a problem with winter survival. We definitely had some creeks open up in our plots and caused a lot of variability from plot to plot. Didn't have the same number of plants. Overall though, we got some higher yield though with 25 R40 compared to AC Samson. I'm disheartened to see how low my yields are compared to the network. We got some work to do with our agronomy, I think. We didn't detect any statistical significant difference in any fusarium factor we're looking at. But we did see low levels of fusarium, we did see low levels of DOM, but the treatments didn't cause any reduction. And if we compared that to what the model actually predicted, the model didn't say we were hitting high risk. It said actually only a 24% chance of an epidemic, so that corresponds pretty well. Now we're getting into spring wheat. This is where it gets more fun. For spring wheat, this is all 3 trials combined. I thought this would be interesting that the fungicides actually increased the yield. So every time we sprayed the fungicide, we got a nice little yield boost of about 10 to 15%. We also, our biocontrol we found didn't give a yield boost. It actually caused a little disease and actually dragged the yield down a little bit. Not what we were looking for, but that's often what happens when you're testing these things early. So for the first trial, the April 16th planting, this is the one with the clearest DON results. We got, in the untreated control, we had over 1 part per million DON. Both the fungicides actually reduced DON a little bit, but the only one that was significant was Miravis ACE. Really exciting though, the biocontrol actually reduced DON a little bit. So it might have dropped yield, but it reduced DON. So I don't know, there might be an application. How would this compare to the maps? Come on, machine. Well, the map said that there was a high risk there, and we did get over 1 part per million DON, and that's what we consider to be, anything over 1 part per million DON is what we consider to be a moderately high level of infection. So when I get into the next 2 wheat trials, this is where the results get a little bit more confusing. In the mid-trial, we had no clear fungicide effects. We did see a difference between the DON levels in Easton versus AC Walton, where there was actually more DON in Easton than Walton. And if we say that those levels are approximately 1 part per million and the risk level was identified as moderately high, that's actually pretty accurate. In the next trial, this is the fun part of pathology. So we got a good amount of Fusarium head blight in exactly 1/4 of the trial. In 1 rep in the trial, we had a lot of Fusarium head blight, and in the other 3 reps, we had absolutely none. And explaining these things, you can blame it on wind patterns or the location in the field or crop residues that were left, but it's often really tough dealing with this. So there's no stats on this. This is just one dot's one cultivar, one dot's the other. And you can see that in that 1 rep, when you sprayed the fungicides, you got the DON below 1 part per million. And if we compare that to the risk prediction, once again, we're at that high risk there, and it was potentially accurate there. But I want to basically say that this isn't a great result. Now, it was high risk for that 1/4 of the field, but it was actually pretty low risk for the other 1/4 of the field. This is where the risk actually comes in as a percentage. So it doesn't necessarily mean that it's predicting that every field's going to get hit evenly, and you're always going to get these patterns of disease in fields where you might have patches that are higher or lower. So going back to that mid-trial where I said there was no clear treatment results, well, this was one visual result we saw. This is actually 3 weeks after spraying, and I hope it comes out there where you can see that actually where we sprayed with Prosaro versus control, the flag leaf stayed quite a bit more green. When he sprayed with Miravis, it got way more green. The flag leaf stayed green for about an extra 2 weeks. You can't see it from this picture here, but the biocontrol actually does look worse. It did cause some leaf infection. If we look at the DON, if we look at the DON results, this is for the 2 cultivars, AC Walton and Easton. When we sprayed with Miravis Ace, we actually had the highest DON. And why do we think this happened? Well, we're still working on that, but we think it might be because there was so much rain during this anthesis period, we had to squeeze in a fungicide application when we could. And it was late, and it rained about 6 hours after we got it on. So we don't think we got the full effects of the fungicide, and we got it on late. But I think this kind of makes sense to me more today than it has over the last month, because after listening to the Yen panel, I think that if the fungicides aren't getting on great, they're not doing the job of controlling FHB and DON. Interesting, Prosaro applied at the same time, didn't see that effect. So not all these fungicides are equal. They all have their own unique properties. So moving on to barley, same thing again. This is the 3 trials combined. We saw Miravis Ace, a little bit of a boost in yield compared to untreated control. Our barley actually looked pretty good this year. There was really little to no DON. In fact, in one of Emily's trials, this is the head she found with Fusarium. So she managed to find this seed was infected with Fusarium, and she cultured it in the lab and confirmed that that's Fusarium head blight. So out of 1 trial, she found 1 seed. We didn't really detect any DON in these trials. So how did that compare to the risk models? Well, using the barley risk models, it actually predicted that there would be low risk below 25% for each of the trials, and that corresponds with what we saw. We actually had relatively clean barley. Now, that being said, the reason I've mentioned Kelly Turkington is we're running some of his barley trials, and they were actually planted at a different time. And those trials we did find Fusarium in, and we are crunching the numbers right now to see how well those trials actually corresponded with DON. And we're also going back in time and looking at every single trial we ever planted going back to about 2010 when the program started to do DON analysis. So to summarize, 2021 was a year with moderately high FHB pressure across the Maritimes. Many fields were infected, but not all of them were. That's just the nature of it. It wasn't as high as it could have been. It could always have been worse. More rainfall and higher temperatures would have made it actually more disease pressure. I want to stress that modeling is really valuable, but it does not replace good crop management. Particularly having good crop rotations where it's not continuous cereals, managing corn residues. Managing is my code word for tilling the corn residues and not leaving them on the surface of the ground. And choosing resistant cultivars when it's possible. If you're growing durum wheat, it's not possible. Our preliminary results showed that the models are pretty accurate, and I think that the risk models we showed today are actually a promising first step. And we're working on a web tool, and we're hoping to work with the Atlantic Grain Council on an extension to this project so that we can get something that can be available on the web for all the producers in the region. Yep. Yeah, a negative effect. And it's a very significant negative effect versus every other treatment.
And so how can you explain not doing the job as opposed to doing the bad job?
With one trial, I can't draw much conclusions of it. We're definitely going to keep spraying Miravis in trials in the future and see if there's a trend. And we're going to look at throwing on some late applications and seeing if that has an effect. But we need to look into this a little further. Trust me, when I first saw this result, I thought that we made a mistake with how we labeled our plots in the field. And it wasn't until literally Emily showed me a correlation between the yield data and the protein with all the expected effects of Miravis with the pictures that I actually kind of took it as face value. Yeah, because compared to the untreated, like doing nothing was below 1 part per million done. Spraying a biocontrol was a little bit higher, but it didn't really hurt other than yield. Prosaro increased yield, didn't really help for Dawn. And Miravis, it went, the highest plot was 15.5 parts per million. Sorry? Sticker. No. Yeah, but if we kind of go back a little bit, we know that Miravis still had an effect because we can visually see it. But definitely, I'd have to check with my technician to know exactly what he applied and how he applied it, but that's something I'll definitely check with him and ask him.
Can you explain the cumulative, if I was to go on the site at a date and it says that this is it's moderately low or something, how is that as of that date, or is that a cumulative effect from first?
So what this model here would be doing, say let's just use the example of Queens County. It's taken the weather station data and then it's looking at the past 7 days from that date and looking at the relative humidity and temperature and then calculating the risk based on the number of hours with relative humidity above 90% and the temperature above 9 degrees. So essentially what it's doing here is every time you calculate it for the day, it's that risk at that day. And then the next day, of course, you're taking 1 day off. So it's not cumulative, and it's literally just the risk that was calculated on that day. So if a producer wants to spray fungicide in a day, I see that they could check the map to see whether it's required on the day or a few days leading up to it. Because I don't expect that with the risk models, all of a sudden you're going to get 1 day happens and the risk. If it's dry for 6 days and then you get 1 day of rain, that's not going to turn it into very high risk.
But if I've had 3 weeks of high humidities, great dews and everything, and then when I go and check, and we've had 2 or 3 days, nice, dry, low humidity weather, have I not still built up a population potential
Well, essentially the population that's there that's ready to do the inoculum, it's a little different. It's actually down on the bottom of the ground. It's got a different climate. And you can pretty much guarantee, except for maybe 2020, that there is enough humidity in its little microclimate zone just from soil moisture evaporating. So really what this is predicting is are the conditions conducive for the disease to develop today? And since the spores have to be put on as protectants, I wouldn't want to say put it on, apply a spray just in case, unless you could see that, yeah, risk was really building up. But Fusarium hebelite doesn't start in the field in the last 7 days. It starts in the field the previous year.
So do any of the other models, the U.S. models, have additional data into it that you can look at and say, okay, last year was bad?
So the U.S. model actually, this is one of the U.S. models. It's actually a really good one. The U.S. model is, what they actually do is, we took one factor out, which is crop history. We took the crop history out of our examination because unfortunately we don't have crop history for all of our sites. When they mean crop history, that's corn. And did you plant corn last year, the year before, or the year before? And if you planted corn in any of the last 3 years, all of a sudden you could take your low to a moderate or your moderate to a high. If you planted corn last year, you might go from a low all the way to a high. Because the corn residue being in the field is really a major factor. But of course, the corn residue is only infected with Fusarium if the conditions in the last year were conducive for Fusarium to develop too. So the modeling can get quite daunting, and what I'm kind of interested in trying to do is have a model that first actually incorporates in the ability of a grower to put in which cultivars they're planting with a risk score of its susceptibility. So, for instance, Durham wheat is going to be a lot more susceptible than, say, AC Walton. AC Walton is more susceptible than some of the newer lines that have come out of the breeding programs like AC Fairwinds. So that would help essentially refine the model a little bit. And that's one of the ways the barley model works. The barley model is going on the assumption that barley's got a little bit more innate Fusarium resistance than spring wheat does, primarily because barley is resistant to the spread of Fusarium. Fusarium doesn't spread from seed to seed. It has to infect every seed individually in barley. Whereas in wheat, one seed, conditions are right, the whole head can be gone.
More often than not, I get a question from a customer who will say, well, I had wheat in this field last year. Should I be putting wheat in it this year? Or,
no.
This is my rotation.
Just my opinion as a pathologist, you should always rotate to non-host crops if you can. I think every rotation that's varied is good. Soybean plants where you have any continuous soybean, you run the risk of building up white mold in the field or root rots, all sorts of different pathogens. There's a whole bunch of benefits to actually not going straight from corn to wheat. But I'd say there's some exceptions for that, though. If you've had a really great corn growing year and you can get your corn off early enough and you didn't have a lot of infection in the corn and you can get your winter wheat in, especially if you're not planning on harvesting that winter wheat for grain, you're using it for silage, the risk isn't there anymore. So there's kind of no one size fits all, and that's kind of where I'm hoping that the web tool will basically be able to provide guidance to a range of growers under a range of different conditions. Just like a weather forecast would if someone's planning a barbecue versus someone planning a wedding versus someone planning a beach day, or someone like me who's going to go home and write a paper in a dungeon or something. Any other questions? If not, I just want to thank everyone who's on this here, which is this is a list of my research team and the groups who work closely with the Harrington Research Farm, and they've done most of the work here, and I need to give them pretty much all the credit for everything that I showed today. Thanks for your time, and I really appreciate it. I'm really happy to see people all in person.